hypothesis, and he borrowed that title from Lewis Carroll's Through the Looking Glass, where the Red Queen said to Alice that it takes all the running you can do to keep in the same place. And when we apply this to the interaction between predators and prey, what it means is that there's no predator that's so proficient at catching prey that it doesn't at times have the risk of starving to death, nor any prey that is so proficient at evading predation that it is immune to the risk of being eaten. It's a constant race between predator and prey that never ends. Now this idea builds on a more general argument that was made by Charles Darwin. In Chapter 3 of The Origin of Species, he talked about the struggle for existence. And what he argued in this chapter is that much of what we see and we attribute to evolution by natural selection is the consequence of antagonistic interactions between organisms, between predators and prey, between disease organisms, or parasites and their hosts, or between competitors for resources. But it's these biotic interactions, these interactions among different organisms that Darwin argued are the most important factors in shaping how organisms evolve or adapt to their environment. Now, what my career goal was, going back into the 1970s, was to take some aspect of this more general theory of evolution and to go out into nature and to try to look at it as an experimental scientist, to be able to have a hypothesis and then to be able to test the hypothesis by making a manipulation in nature and watching organisms evolve and seeing if they evolved in a predicted fashion. And in the 1970s, it was a radical proposition, because at that point, we didn't think that evolution was fast enough to be an observable process. And yet, what I just learned in my population genetics classes suggested to me that in fact it was, and this is something that was feasible. Now, the particular aspect of evolution that I chose was then a very popular theory of life history evolution. And I chose it because it incorporated a generalized concept of fitness. To Darwin, fitness was the ability to survive. If an animal was able to survive for a longer period of time by eating more prey or by not being eaten by a predator, then it would have more opportunities to produce offspring and be represented in subsequent generations. Life history theory took a more general and mathematical perspective, and it argued that fitness wasn't survival, fitness was reproductive success. It was how many babies did you produce during your lifetime. And what was interesting to me is that it could do seemingly counterintuitive things. It predicted that under the appropriate circumstances, natural selection could favor the evolution of a shorter lifespan, or natural selection could favor the evolution of producing fewer offspring. And the reason this could happen is because there are trade-offs or relationships among these different things, and those changes were compensated for by other changes in the life history of the organism. So it was really the end product, how many you produce, that was important. But the more important thing about life history theory is that it yields testable predictions. And what I wanted was a theory that made a prediction, and that enabled me to go into nature so that I could design an experiment to test that prediction. The components of the life history are things like how big you are when you're born, how old you are when you begin to have babies, how many babies you have, how large they are. Do you have many that you devote little to each of them, or do you have a few and devote a lot to the ones that you have? And also, how often do you reproduce? And then at the end of the life history, there's also issues that are like senescence and aging and menopause. But all of these are the components of your life that contribute to how many offspring you're going to be able to produce during the course of your lifespan. Now, to understand how the theory works, you need to think of your life as being like this pie. And the idea of the pie is that it's a finite size, and you divided it up into different slices. One slice of the pie is maintenance. Most of the food you eat, most of the resources that you're consuming go to maintaining your body. And they include things like maintaining your metabolism. We're all warm-blooded and our metabolism eats up a lot of energy. But they also maintain the parts of your body that are constantly wearing out and being replaced. So for example, your blood cells only last for a few weeks. And so you're constantly making new blood cells, you're constantly making new skin, you're constantly replacing the lining of your intestinal tract, and there are other parts of your body that are quickly wearing out. And so maintenance goes into constantly replacing them all. The second component of your life history is growth. Most of us have stopped growing, but when we're younger, a lot of the resources that we consume are fueling growth. Some animals will grow throughout their lifespan, and so there's a constant capacity to adjust how much you put into growth versus other things. A fourth slice is storage. We don't often think of fat as being adaptive, but that's usually because we make more deposits than withdrawals. For many organisms, during the course of their lives, fat is very adaptive because it's the capacity to establish a savings account during times when food is abundant so that it can be spent later at a time when food is scarce. And so, for example, right about this time of year, or the next couple of months, black bears in the northeastern United States are going to go into hibernation. And while they're still in hibernation, they're going to do the most expensive thing in their lifetime, which is that the mothers are gestating, they're going to give birth, and then they're going to lactate. They're going to provide milk to their offspring. And they do all of that without feeding. It's while they're still in hibernation that this is happening. And the reason they can do it is because during the previous summer, when there was a lot of food, they stored a lot of fat. And that fat is being used at a later time when it's a more favorable time to produce offspring. Then finally we have reproduction, and that's the size of the slice that goes into actually making babies and supporting the growth of babies. But what life history theory does is to talk about what the best way is to divide the pie up among these 4 different slices to result in your producing the largest number of successful offspring during the course of your life. And so that's the theory that I work with. Now here's a sample prediction from life history theory. The prediction was that if you're an animal that lives in an environment where there's a high risk of being killed by a predator, the natural selection is going to favor those individuals that can begin to have babies as soon as possible at an early age and that invest a lot of their resources into reproduction. And in doing so, and starting young and putting a lot in, they're actually taking away what they could have for the future. A consequence of this kind of life history might be that they aren't going to maintain themselves as well, and they aren't going to be able to live as long as if they had put more of their resources into growth and maintenance. But the fact is, they're not going to live long anyway because there's something out there that's going to eat them. And so this is the division of resources that's the best under this particular set of circumstances. Now, one of the reasons I chose this example is that what it's saying is that the anticipated effects of predation go far beyond camouflage and defense. Predation can actually shape how your whole life is put together. Okay, I work on guppies. You all know about guppies. This is a male guppy and a female guppy. And the first thing that you'll notice is the strong sexual dimorphism. The males are very brightly colored. The females have no real coloration at all. The males are also highly variable in coloration, and those colors are used as part of displays, sexual displays, to show off the females and try to induce females to want to mate with them. The males are smaller than females. The male is about 15 millimeters long. An adult female can be 20 to 25 millimeters long. They're live bearers, and they have rapid development time. So the time interval between when a female is born and when she first gives birth to her own babies can be as little as 10 weeks. And then she'll give birth to babies about every 3 to 4 weeks thereafter for the remainder of her life. And the babies are like miniature adults, and the way you keep them in the laboratory is very much like the adults. All these combinations of attributes means that guppies are a great animal to work with in the laboratory, but as you'll see in a few minutes, they're also a great animal to work with in nature. And so they're one that's suited all of my different goals for research organism. You know all about Trinidad. The main point about Trinidad from this map is that Trinidad is a continental island. It's a chip off of South America. And a consequence of that is that the flora and fauna here is a subset of what you would see on South America. But on the other hand, a consequence of being an island is that the biodiversity is lower, and in some regards, features of the environment are much more repeatable and predictable here than they would be on the mainland. And I'll return to that point in a minute. I do all my work in the streams of the Northern Range Mountains. And usually when I'm in North America, I'll explain to people that this is a large river by Trinidadian standards, a small creek by North American standards. But also the important point is that this is a rainforest that gets on the order of 3 to 4 meters of rain throughout the year, so it's well supplied with streams that will flow throughout the year. And that's the habitat where guppies live. But the other thing I'll tell them is that I grew up in New York dreaming of seeing the tropics. I knew the tropics through things like National Geographic specials on TV, and I can remember well sitting in the den while ice was building up on windows, looking at this beautiful tropical scenery and the remarkable wildlife that could be found there. And I decided then that this is something I wanted to see during the course of my life. And so the guppy research has really been my ticket to the tropics for the past 30 years. Here, what I'm interested in Trinidad, what brought me here to begin with, was work that other people had done, which already described the kind of menu of circumstances that I needed to do the sort of science that I had in mind. And what had been described by prior researchers is that you could go to streams in the northern range and find guppies in these two types of settings, in what I refer to as either high or low predation communities. In high predation communities, guppies live with fish like these. This is the wolffish. You may all know these animals. This is the top predator in these streams, and it's an animal that can really hurt you. This is a fish that very much likes to eat guppies. It's the pike cichlid. And a guppy in a 10-gallon tank with one of these fish has a half-life of less than a second. But it turns out that in nature, guppies are very good at evading these predators. And so a fish like this eats, we think, on the average, about a half a guppy to a guppy per day. But that rate of predation is enough for them to have a profound effect on the evolution of guppies. And this is what's locally called a sardine. It's a charasin, and it also is probably a serious guppy predator. If you move upstream, all of these fish disappear, and you can find streams reliably that just contain just guppies and this fish, which is locally called the jumping wabine, and it's a killifish, which is found all throughout even the smallest tributaries in the Northern Range Mountains. What you're looking at here is a schematic of the Northern Range, so that the mountains run on an east-west axis, and then you have rivers that drain either the south slope or the north slope. And then color-coded on each river is the distribution of high predation localities in red and low predation localities in blue. But the main point here is that each one of these rivers presents you with the same contrast. Over and over again, these rivers have high predation localities in the lower portions of the stream, and then they yield their way to low predation localities, either in tributaries or in the upper portions of streams, because the predators are excluded by rapids and waterfalls, but the guppies and the killifish are not. From a scientific point of view, this is as if you could think of the Northern Range Mountains as being like a natural experiment which is naturally replicated. We have many streams that we can go to and look at guppies that are found with predators, and then we can look at very closely related guppies that are found without predators, and we can use genetics to know that the adaptation to life with and without predators happened over and over again in some of these different river drainages. And so it is a truly replicated, or it presents a truly replicated opportunity for studying the process of evolution by natural selection. Another feature of many of these streams is that the border between high predation and low predation localities is sometimes a very abrupt one. It's defined by a waterfall. So, for example, you can find settings where you can find guppies with predators below a waterfall and guppies without predators above a waterfall, and they're within tens of meters of each other. And it means that the differences between those environments are not correlated environmental differences. It's the same river. It has the same nutrients, it has the same temperature, it has the same light availability, the same chemistry. The only difference between above and below the waterfall is whether or not the predators are there and other correlated things that the predators cause to their environment. Okay, the work that I did is divided into different components. And the first component I'll be talking about is my study of demography. What I wanted to know is, what is the death rate of guppies that live with or without predators? It may make common sense that if you live with a predator that will eat you, that your mortality rate will be higher. But for various reasons, it was important to actually measure the mortality rate of guppies in nature and to find out what the nature of the differences were in the risk of mortality in sites where they lived either with or without predators. And to do this, we became guppy tattoo artists. Originally, what I did was to take acrylic latex paints and dilute them in a teleos ringer solution to reduce the viscosity, and then would inject little dots subcutaneously in the layer between the scales and the muscle so that we could recognize them as individuals. But then a company came along and actually invented a product for exactly that purpose. What they did was to take an elastomer that was developed for human surgical implants, so it's hypoallergenic. Then they colorized it and then sold it for fantastic amounts of money to people like me who wanted to do mark recapture studies on fish and other kinds of aquatic organisms. And right now we're working actually with 10 different colors, and there are 8 different positions on guppies where we can mark, and we have 2 marks per fish. Now, if you multiply up that combinatorics of 10 colors times 2 marks times 8 different places to mark fish in all different combinations, what it means is that we can mark thousands of individuals and recognize them as individuals in natural populations and follow the fate of a fish that we know, that we can name, as opposed to just being able to recognize them as a guppy in a stream. What you're looking at here are some of the accommodations that we have when we're doing extended mark recapture studies. And this is an invention, a Hennessy hammock, that changed my life for doing field work because it's a completely enclosed sleeping unit. It has a built-in mosquito net and rain fly, and it has you up off the ground. Because I discovered early along in my research in Trinidad that sleeping on the ground in the tropics can sometimes invite unwanted bedfellows. So it was nice to sleep off the ground, but it's also a lot easier on your back when you get up in the morning. But the next thing I found that surprised me is that the tropics can be really cold. You don't think of sleeping in a rainforest overnight at 70 degrees as something that would require a blanket, but it turns out that if you have 70-degree air flowing on all sides of you, it's cold, and you have to bring a blanket to the field, which I do now. I didn't do when I first did it. Okay, this is the kind of stream where we can do our best guppy demography. Many of your northern range mountain streams have what we call a pool riffle structure. So what you're looking at here in the foreground is a pool, and that's an area of the stream where there isn't a linear current from the top to the bottom. Instead, water flows into the pool, and then you have patterns of turbulence on the inside of the pool, and then at the bottom of the pool, water flows out. And then the riffle, which bounds the pool on the downstream and the upstream side, is an area where the water is flowing in a straight line from the top to the bottom. And so the stream is structured like a staircase. So you have a pool, and then a riffle, and then another pool, and a riffle, and then another pool. And what I discovered early in my career is that guppies like to hang out in pools, and they're disinclined to swim from one pool to the other through a riffle. But I also discovered that you can catch every single guppy in the pool without ever setting foot in it and without disturbing the habitat. And the way we do that is we can hold a butterfly net in each hand and you stand at the top end of the pool and perhaps stir up a little bit of sediment. Now, if you did that, most fish would run and hide. When you do it in the guppies there, it's as if you created a guppy magnet. They're all going to come swimming towards you because they want to see if there's anything good to eat in that sediment that you just stirred up. Then you can play them from one net to the other and pull everyone out. And as I said, you can do that without ever going into the pool. Many of the ways that we collect fish are kind of destructive to the habitat. And if you want to visit the same habitat again and again, then there have to be ways of doing it without disturbing the habitat. So we're able to do that with guppies. It might take me about 45 minutes to catch every guppy in a pool like this, and there might be 80 to 120 of them. And what we'll do is keep them all in one collection container in medicated water, bring it back to the laboratory. We will measure every single guppy, we'll mark them as individuals, and then we'll return them to the pool of capture. And we treat each pool as a unit of observation. So we'll keep this pool of fish separate from this pool and this pool. We'll return them to the site of capture, and then a month later we'll come and catch them again and pull them all out and collect the same kind of information. Now, if you put that kind of information together for a succession of months, then you can learn a great deal about the fish. You can know about individual growth rate, because you can measure fish every time you catch it. You can know about mortality rate. The fish that were there before but aren't there anymore are ones that likely died. You can know about birth rate. Birth rate, the new recruits, the newborn, are the ones that are unmarked and small, that appeared in the time between the two different census periods. And if you put all that information together, then you reconstruct what's called the life table of the population. You can talk about what the probability is of survival from one time period to the next, what the growth rate is, and what the net birth rate is of the population. Okay, what you're looking at here is the very first result I got from doing mark recapture. And this slide actually represents 14 different experiments, 7 each on a high predation versus low predation environment. Here you're looking at the probability of survival, and here you're looking at different size classes of guppies. The main point here is that the probability of survival if you're a guppy in a high predation environment is significantly and substantially lower than if you're a guppy from a low predation environment. Now this just represents the probability of survival over a 12-day time interval. What we've done now, what that data implies is that if you continued for 6 different months, that the probability of being alive after a 6-month period, if you're from a low predation environment, would be 20 to 30 times greater than if you're from a high predation environment. We've now done these 6-month studies, And we've shown that these very short time intervals are accurate predictors of long-term differences between guppy populations from high and low predation environments. So the first conclusion is that guppies that live with predators have much higher mortality rates than guppies that don't live with predators. But we learned other things as well. We learned that they grow much faster and they have a much higher rate of recruitment of new babies into the population. Okay, the second thing that we did was comparative studies. I wanted to know what exactly is the life history like of guppies from high predation environments that are represented here by circles and low predation environments that are represented here by squares. And so we sampled guppies from a large number of streams, and very often we had paired comparisons between a high and a low predation environment in the same stream. Then we took them, these were animals that were collected in the field and preserved, and we took them back to the laboratory and dissected them and characterized what the life history was. Then the other thing I did was experiments in the laboratory. And in this case, what I would do is to collect an adult female from the field, actually a sample of adult females, and bring them back to my laboratory, first in Pennsylvania, then in Maryland, now in California, and put each adult female in her own aquarium and take advantage of a handy feature of guppy reproductive biology, which is that adult females store sperm and they're always reproducing. And so it means that every single female that I put in a tank of her own would produce a succession of litters of babies. Each of them from one female became a numbered lineage for future experiments. And then I would rear them through 2 generations. I had the wild-caught mothers. They had babies, which I raised to maturity, and then I crossed, and I had a special crossing design that retained all the genetic diversity of the original population and equally represented all of the original females that were in the collection. And I took their offspring, which are now the grandchildren of the wild-caught females, and compared their life histories. Now, the reason I carried them through 2 generations of the common environment is that I wanted to conclude that the differences in life histories between guppies from high and low predation environments were likely to have a genetic basis. And one of the sources of differences between them could be that the mothers developed in a different type of environment. It turns out that the mother's environment can influence features in the babies. So what I did then was to create a generation of mothers in the laboratory so that the grandchildren all had mothers that grew up and developed in the same environment. So the idea was to control the environment of the mother so that I could infer that the differences between the babies were ones that were almost certainly genetic differences between them. In some cases, I also did hybridization studies to look at the genetics of the differences. So this is the kind of laboratory where I did that work. When I was looking at the grandchildren, I would rear 1 fish in a 2-gallon aquarium. I fed them quantified amounts of food. I had Hamilton micropipettes and could measure their food volumetrically to the nearest half microliter, which is a teeny tiny amount. And so I knew exactly how much food I gave them, and I knew exactly how much food they ate, because guppies are very pliable. They like living in the lab, or at least they take to it very well, and they'll swim up. When you walk into the lab in the morning, they're all right at the front of the tank watching you, waiting to be fed. When you put the food in the water, they all eat it before it hits the bottom. So what that meant is I could start with the fish at a very early age, and I could know exactly how much food went in. I knew how big the slice of the pie was, and then I could measure what they did with that food. I could measure the size of all the different slices and ask if the slices produced by fish from a high predation environment were different from those from fish in a low predation environment, and if the nature of those differences were consistent with what theory predicted. And the answer is that it was. Both my comparisons of wild-caught guppies and my laboratory common garden experiments show that there were dramatic differences between guppies from these 2 different types of environments, and that those differences were consistent with what theory had predicted. This is a cartoon version of the differences that I saw. The guppies from the high predation environments were much smaller and younger at sexual maturity than those from low predation environments. They produced many more babies per litter, and the individual babies were much smaller than the babies produced by those from low predation environments. I also showed that these animals had a much higher amount, the slice of pie that was devoted to reproduction, which was much bigger in these fish than in these fish, for 2 reasons. One is that they put more into each litter of babies than these fish did, and the other was that they had babies more frequently. The time interval between successive litters was shorter. This is the only data slide I'm going to show you, and it shows you where the numbers came from in the slide that you just saw. The first number here is how many babies each female produced, and the second number is how big the babies are. In red is a high predation locality. This is the Aripo River. And then in yellow and blue are low predation localities. The point is that all low predation localities have females that have about 3 babies per litter, 2 to 3 per litter, and the babies weigh about 1.7 milligrams. Down here in the high predation locality, now you have 7 babies per litter, and the babies weigh about 0.8 of a milligram. So that's what you saw in the previous cartoon. But the interesting thing are these two sites. They're from just above and below one of those barrier waterfalls, and the differences in those guppies that were found only 50 meters apart in habitat that was identical in all regards, save for the presence or absence of predators, are almost as dramatic as the differences that you would see far downstream and far upstream. The one neat thing is that these guys just below the waterfall are kind of intermediate. They produce somewhat fewer babies and somewhat larger babies than you would see about 400 meters downstream. And we know now, using genetic information, that this almost is certainly a consequence of gene flow, the movement of individuals from above the waterfall to below the waterfall. And we have genetic markers now that show us that that kind of movement is taking place. But by the time you get to 400 meters downstream, the effects of predators are so intense that the influence of that gene flow, that individual movement, disappears. Now this is just what the guppies look like from a low predation site and a high predation site on the Aripo River. The males on the high predation site are smaller and younger at maturity, and the main point is that these are differences that you can see. The differences in life histories between guppies that live with and without predators are not subtle. They're of the same order of magnitude that we very often would associate with differences among species. And they extend to characters much more than life history traits. I'll give you a summary later on of all the different ways in which predators have changed guppies. Okay, the conclusion then is that, as predicted, the guppies that live with predators are younger at maturity, and they devote more resources to reproduction than guppies that live without predators. And remember that this was a theory. It was a theory that predicted that this is what should happen as animals adapt to a high risk of mortality. We showed that the high risk of mortality is there, and we've shown that the differences, the predicted differences in life histories are there. But what I had in mind was more than that. I wanted to do experimental evolution. In other words, I wanted to go to nature and actually manipulate the environment and see evolution in action, and in that way test a particular theory. And so everything that you've seen up until this point just sets the stage for attaining what my real goal was. And what I've done is to actually increase or decrease the risk of mortality in natural populations of guppies, and then I've quantified their subsequent evolution. Okay, here's a schematic of distributions of predator types in a hypothetical river that illustrates the kind of experiments that I did. Very often what you find in nature is a barrier waterfall below which you can find guppies with predators, and above which you have a guppy-free environment that contains the killifish, the jumping wabi. And so in this kind of setting, the experiment that you can do is you can take guppies that live with predators down here and introduce them over the barrier waterfall and allow them to extend their range upstream. And so you've moved them from a high risk of mortality to a low risk of mortality, and you can evaluate whether or not these guppies evolved in the predicted fashion by comparing them with their ancestors collected from below the barrier waterfall. This is an experiment that actually was initiated before me by John Endler, who was interested in looking at the evolution of other features of guppies and how they're shaped by the presence or absence of predation. And then it was one that I repeated once more in the second tributary. Then there are other ways that you can increase the mortality risk of guppies by introducing a predator into a small section of stream that's bounded by waterfalls. Okay, what you're looking at here is my first very big result from those experiments. These are experimental guppies. These are guppies that were collected from a high predation site below a barrier waterfall and introduced into a previously guppy-free low predation site above the barrier waterfall by John Endler in 1976. And then I looked at them in 1987 and asked whether or not they were genetically distinct. And what I found is that the male age of maturity was significantly greater than those in the control site. The male size of maturity was significantly greater than those in the control site. I also found that the females were older and larger at maturity, and that they were producing larger babies, and that they were investing more in their first litter of offspring, but not in their second and third litters of offspring. And that actually is an important detail that matches the predictions for how people think that life histories will evolve, because they predicted that what you do early in life will have a more significant contribution to fitness than what you do late in life. And so what I showed is that they had evolved. Now, many people looked at these results and said, okay, they're statistically significant, it happened. but are they important? And the answer that I'll give you a little later on is that, yeah, they are important. This actually is a very striking and remarkable result. It also brought me my 15 minutes of fame, my Andy Warhol 15 minutes of fame, because even though I thought it was a good paper, I didn't think that much of it. And I found myself on page 1 of the New York Times and the World Herald Tribune for this result, because it was the first time that somebody had tested some aspect of the theory of evolution in a natural setting with an experiment and shown that the prediction was upheld. Okay, so the third conclusion then is that transplant experiments show that guppies do evolve as predicted, but the other thing is that they evolve very quickly. I got some significant results in as little as 4 years, and that was much sooner than people anticipated. When I told people I wanted to do this kind of work, very often they would smile and say, "It's a great idea. We hope you live long enough to see something happen." And the answer was I didn't have to wait that long, and that was a surprise. Okay, what I want to do now is talk a little bit about what I do in Trinidad when I'm not chasing guppies. Because my real ulterior motive for coming to Trinidad, and what really got me in biology to begin with, was my love of amphibians and reptiles. And what I wanted to see in the tropics, and what my attraction was in the first place, was that the tropics are the land of superlatives. It's where you can find the biggest or the smallest or the most beautiful or the most dangerous of anything. And so this is a series of photos of what I found when I was not chasing guppies. When I first came to Trinidad, whenever I was done with the day of working on guppies, I would go out at night and look for reptiles and amphibians. And the compelling thing about doing herpetology in this kind of environment is that what you find in the morning can be different from what you find in the afternoon or the evening or at night. And so there's no incentive to ever go to sleep if you want to find new things. And for my first several years here, I pretty much worked myself to exhaustion, trying to be out at all times of day in all habitats to see what I could find. I've gotten over that now, but like I said, these are my tropical superlatives. So this is the smallest. This is a fully grown, sexually mature ashy gecko on my forefinger. This is a coral snake. It's the most dangerous. The first was the smallest. This is the most dangerous. Some of you probably know that the coral snakes produce a neurotoxic venom, and that where fertile ants can make you wish you were dead, a coral snake can really kill you because of the toxicity of its venom. But if you're a herpetologist, they present a herpetologist dare, which is that even though we know they can kill you, we also can observe their head and recognize that the head is too small for them to really bite you, and so the dare is to pick them up. This is not my hand. This is the rarest. This is the golden tree frog, which is only found on a few patches of the tallest mountains in Trinidad, where you have a patch of cloud forest. And a member of this audience found one of these a few weeks ago. It's a rare thing to ever get to see them. But the point is that they have a very tiny range. They're highly endangered for that reason. Their closest relatives are found in similar habitats in Venezuela. And what we think is that where they are now represents a sky island. That at a time when the world was cooler and wetter, these animals probably were broadly distributed in the lowlands. But then as the world got warmer and drier, their range contracted so that now they persist only in these small sky islands where some cloud forests persist. This is the largest. This is a bushmaster that I found on Blanches's Road. It was a male who was 8 feet 2 inches long. I'll tell you later how we know that if you really want to know. It's one of the high points of my career as a human being to be able to see one of these things. This is the most beautiful, my nomination for the most beautiful snake. This is a rainbow boa, but it's an Ecuadorian rainbow boa. The Ecuadorian ones retain a very bright coloration into adulthood. So this is a female that's about 7 feet long. And she's also pregnant. We sat and looked at her and thought, each baby is worth $300 on the pet market. We could pay our whole field season with her. We let her go. She was the nastiest snake I ever met in terms of being one of the most beautiful snakes I ever met. Okay, what I'm going to do now is talk about what some of the general implications are, or the extensions of my work. You may say, well, it's nice to work on guppies, but what do we really get from the work on guppies? What are the general consequences of that? And the first consequence I'm going to talk about is what we've learned about the rates of evolution. What you're looking at here is a comparative table on inferences about how fast evolution has happened, looked at in different types of data. And the unit that was used here, this number, is something called a Darwin. And the Darwin has a property which is that you can look at the change between 2 points in time and ask how fast did that change take place. And it's a metric that can be applied to data from many different sources, so it has some handy properties. But if you look at the fossil record and you look at what people would call rapid evolution in the fossil record, the rate at which you're seeing evolution happening is about 1/10 of a Darwin to a few different Darwins. If you look at evolution in colonizations, and these are mostly historical colonizations where people were mostly Europeans spreading throughout the world, took European animals and introduced them to other parts of the world, then we've seen in many cases that the animals in their new homes are different from what they were before after a couple of hundred years. So we've seen that those rate of changes are in the hundreds of Darwins. These are how fast organisms can evolve in laboratory selection experiments. So when people looked at how fast this was relative to how fast these things are, the reaction was, well, so what? This is the laboratory where you can impose as intense selection as you want. It doesn't represent what's going on in the real world. These are the rates of evolution that I saw in my experiment. The rates of evolution of the male traits were actually 25 to 30,000 Darwins. And the average rates of evolution that you were seeing in those earlier studies, my experimental studies of evolution, were about 10,000 Darwins, which means that however small those changes after a period of 10 years may have seemed to you, if you put them on the same scale as inferences from the fossil record, and this fossil record that people had used for trying to understand how fast evolution can happen, but if you put them on the same scale, then the rates of evolution that I saw were comparable to what people could get in artificial selection and were on the order of 10,000 to 10 million times faster than you see in the fossil record. And so what it said is that for all those people who told me that it's a great idea and I hope you live long enough to see something happen, all those people were thinking evolution happens like this. Everyone was looking at the fossil record and thinking that this is the history of life, this is how fast it happens. And what we've discovered since the 1980s is that, in fact, evolution is different. Evolution can be much, much faster than you would infer from the fossil record. And all it took was the will to systematically look for it in nature. And it's very easy to see and document the rate at which this kind of evolution is occurring. Now, one of the things that happened when I first got these results is that I wrote in one paper that this kind of result said something important about ideas that people were proposing about evolution that were based on inferences from the fossil record. For example, at that point in time, there was an author named Stephen Jay Gould who promoted the idea of punctuated equilibrium. And he talked about how when you see episodes of evolution this fast in the fossil record, they're too fast to be accounted for by evolution by natural selection, and that something else had to be going on. And the point that I made is that they're not too fast, they're too slow. You shouldn't try to be coming up with an explanation for these intervals of jumps, because in fact the rate at which things really can change is so much faster. And I said that in an article when I realized was that I would attract unwanted attention, because this was an essay that Stephen Jay Gould published in a very famous series of essays called This View of Life that appeared in Natural History magazine. And he talked about the paradox of the visibly irrelevant. He's a good writer, and so I'll share the first sentence with you. What he says here is that an odd principle of human psychology, well known and exploited by the full panoply of prevaricators, from charming barkers like Barnum to evil demagogues like Goebbels, holds that even the silliest of lies can win credibility by constant repetition. And the silliest of lie that he was talking about here was the idea that evolution in real time is so fast that the rate at which we see evolution happening in the fossil record is irrelevant, that it doesn't really tell us about the rate of evolution. It may tell us about the history of life, that if you want to know about the rate of evolution, you should be looking in real time. And this essay, by the way, was about me. It featured my work. But in any case, the point is that the idea that I was presenting is one that's prevailed. People now accept the idea that evolution really is that rapid. Many other investigators were showing it at the same time in other systems. And it really has changed our concept of the role that evolution can play in real time, and enabling animals, for example, to adapt either when they invade new environments or when they experience or encounter new invaders or other changes in their environment. Okay, here's a summary of the traits that we found evolved in response to life with or without predators. So I talked about age and size of maturity, how many babies animals have, offspring size, reproductive allocation, and lifespan. It turns out that we've shown that the rate of aging evolves as a consequence of life with and without guppies. We've also shown that there's an evolution of morphology. Here's the typical body shape of an animal from a low predation environment versus a high predation environment. And it turns out that these differences in body shape are functional. They tell us something about the swimming ability of the animal, the ability for endurance swimming or acceleration, and also the differences in the shape of the head might be telling us something about what kind of diet they're adapted to. We've seen the evolution of courtship behavior and schooling, and also the evolution of male coloration. What you're looking at here is what the last moment looks like in the life of many small fish. Many of the predators that they encounter, including the predators that live with guppies in Trinidad, generate a vacuum. They have a mouth that is adapted to increase in volume very quickly when the mouth opens, and it will literally suck prey back into their oral cavity. But it turns out that guppies in particular are able to survive this kind of attack. And we've shown that guppies from high predation environments differ in neuromuscular performance. They have a higher response rate, and they can accelerate faster than do guppies from low predation environments. And we've even shown that these traits evolve in response to our introduction experiments. So as I said, the influence of predators on their prey is pervasive. It influences many features of the biology of the animal. One of the things that's in development right now is that we have a complete genome sequence for guppies, and we're beginning to look at the genetics of adaptation. And so all those traits that I saw listed for you, some of which are biomedical interests, like rate of aging, are ones that may someday become accessible to genetic studies. And we can look at the genes that are associated with development rate, or with aging rate, or with neuromuscular performance, using guppies as a study system, because we have these natural populations of guppies that have adapted to life with and without predators, and as part of that process has evolved natural differences in all of these properties of their biology. Okay, another point that we're doing that represents my current research, and this is a grant which expired, but I have a new grant to extend this work, is to do experimental studies of the interface between ecological and evolutionary processes in a natural ecosystem. Showing that animals evolve this quickly can have an influence on how we approach the study of ecology. The study of ecology has generally treated organisms as if they were constant, or not evolving. And the reason it works that way is because we assume that ecological interactions are so fast relative to evolution that we can study ecology and pretend that evolution doesn't happen because it's so slow that it's not going to happen to any practical degree during the course of our studies. We know that's not true, and so it becomes much more interesting to ask how ecology and evolution are shaping each other in real time, or how they're interacting with each other. And that represents the work that we're doing right now. As part of that work, we've now built a series of replicate streams alongside a natural stream. And what we do is there's a spring up here that normally would flow into a stream that's behind this canopy. We divert some of the flow to this tank, and then we gravity feed it through these channels. And we can show that each of these channels generates a reasonable facsimile of a natural stream. And so we can do replicate experiments on guppies, which are different and better than anything that we're able to do in a natural environment. And that's part of the work that we're doing right now. Another consequence of guppy work is that I argued back in the 1980s that guppies could be a model for fisheries, that humans that are exploiting fish as a source of food are like a predator, and they tend to be preying selectively on the largest and oldest individuals in the population. And a consequence of human predation is that commercially exploited fish should be evolving, and they're evolving to become younger and smaller in maturity, which means that they're becoming less useful as a source of food. And people, beginning in the 1990s, have been pursuing this idea and showing that it in fact is happening, that humans are imposing evolutionary changes on the organisms that they're exploiting as a source of food, and as a consequence, they're making them less suitable as a source of food. And this is something that's a new idea that's happened since we began realizing that evolution is that fast. Here's just my result on aging. The point is that the theory of life history evolution has a corollary that predicts how quickly organisms should age, and it has something to do with their life expectancy. And so we tested that theory by comparing guppies from high and low predation environments, and we found, in fact, that they do differ in their rate of aging and in their life expectancy. So it's yet another potential practical application of using guppies as a model system to study biological features that are of interest in human health issues. Then finally, I'm interested not only in guppies, but the whole family that includes guppies. And that's a family called the Pisciliidae, and they're all live bearers. And what you're looking at here is the guppy, but you're looking at sample males from other species in the live bearer family that includes guppies. And many of them are like guppies in that the males are very gaudy and ornamented and have courtship displays. And that's the reason most people have been interested in these fish. But what I've been doing is pursuing a different aspect of their biology that was discovered in the 1940s and for some reason was ignored. And that is that some of these fish have the equivalent of a mammalian placenta. What you're looking at here is a species of fish, and this is a baby early in development, mid in development, and a baby that's near birth. And what you can see is that they're growing remarkably. And what we see when we're looking inside some of these fish is that some of them have tissues that are both maternal tissues and embryonic tissues that are specialized for a mother to transfer resources to the babies during the course of their development. We've now combined these observations with the DNA-based family tree to show that these fish have evolved a placenta at least 8 times, sometimes very recently, and sometimes we have very close relatives to each other that either do or don't have a placenta. And so it's given us the ability to study the consequences of having a placenta in a way that's never been possible in a mammal. All mammals inherited their placenta from a single common ancestor that lived over 100 million years ago, and whatever its properties are today are well removed from the origin of the placenta. And so you really can ask, what happened? What were the consequences of the advent of the placenta? And here you can actually ask it, and you can look at aspects of the genetics and biology of placentation in a way that would never be possible in human beings. So that's another research program that I have going on. And finally, I want to share with you an image of having been bent over a stream, watching guppies for a couple of hours, and standing up and being hot and being dizzy as the blood rushed to my head, and turning around and confronting this snake. Fortunately, I had a camera in hand, so I could capture the moment. Thank you very much.